Good afternoon and welcome to this first of the Dean's Research Seminars for 2021. And today we're very lucky that Associate Professor Helen Billman-Jacobe has agreed to talk to us. Let me tell you a little bit about Helen. Helen's completed her Bachelor of Science at Monash University and then worked on bovine tuberculosis and paratuberculosis at the CSIRO before completing a PhD on recombinant protein expression here at the University of Melbourne. She then returned to the Department of Microbiology at Monash to lead a team investigating drug targets to treat tuberculosis. In 2000, she came back to the University of Melbourne as a lecturer in microbiology and continued her work on TB, attracting substantial NH and MRC research funding. 10 years later, 2010, Helen started teaching food microbiology and biotechnology in the Department of Agriculture and Food Systems and started new research projects on antibiotic-resistant bacteria in food production animals. She transferred to the Melbourne Veterinary School in 2015 to focus on veterinary antibiotic use as part of the National Centre for Antibiotic Stewardship Centre for Research Excellence. Over the years, Helen's trained 44 research students and her papers have been cited over 3,000 times. And since joining the university, she's served on many faculty and university committees, particularly around the areas of graduate research management. In 2019, she and her co-author, Dr. Anne Westmore from the Melbourne School of Population and Global Health, released their book, which was called Breaking New Ground. I remember going to the book launch, and it was a pleasure to meet the people she wrote about, or they wrote about, because they chronicled the experiences of 14 women who attended Dookie College between 1947 and 1965, a time when women studying agriculture was not widely accepted. And it was a pleasure at the book launch to hear the stories from these women that Helen and Anne had captured, and to meet the ladies themselves. Helen's work on crop protection has been a parallel stream of her research, and she's focused on finding microbiological solutions to pest control, novel and innovative work. And in this research seminar, she'll discuss the problems associated with protecting crops from pests, slugs, and snails, and the alternatives to using highly toxic chemical baits. Please welcome Associate Professor Helen Billman-Jacobe. Helen, over to you.
Thank you very much for your introduction, John. Let me just get this going and share my screen. Okay, is that good?
Yep, that's good, and it's perfect, Helen.
Okay then, thank you. So I'm going to talk about control of pests, slugs and snails. So let me first address what are the pests and identify the problem. So we'll start with the pests. The slugs and snails that you can see here are all air-breathing terrestrial mollusks known as gastropods. They're called gastropods because basically their body format is they're a foot that carries around a large digestive system. I'll use their common names wherever possible. Now, all of these species have been introduced into Australia and they're invasive. And they differ for various reasons as to why they're considered to be pests. The ones on the top row are pests in the grains industry. So we've got the black-keeled slug, the grey field slug, two of the pointed snails, and two of the round snails. On the bottom row are molluscs that you actually might find in your garden in Melbourne. Now, some of them damage plants, and some of them are just a nuisance. Now, I actually have to admit I quite like some of them. This little yellow slug has got violet-coloured eyes, and the leopard slugs come in a variety of really interesting markings. I have a number of these that line up each night at my dog bowl, hoping for a free feed. That's just a nuisance. It's some of the other slugs and snails that cause damage to the plants. And it's really control of these plant damaging pests that we have focused on. Now here are some photos that illustrate some of the problems that pest molluscs cause farmers who are raising crops. In the top left, there's a field of canola. So molluscs damage plants, and when plants are young, as shown in this field, they are particularly susceptible to grazing. And you can see that there's large areas of this field where there's no seedling emergence, and this is probably due to grazing by slugs on the seedlings. Some of the pointed snails shown here in the palm of somebody's hand are really small, and the problem with them is that they cause contamination of grain at harvest, and they're right next to some canola seeds. The problem with contamination of grain is the value of the contaminated grain is lower. It can't be used for some purposes, and it might only be able to be sold as animal feed. Slugs and snails also cause crop losses, and some crops are particularly vulnerable, things like asparagus and strawberries. And we've talked to some growers that have lost their entire crop in some seasons. So crop losses in the Australian industry from slugs and snails is estimated to cost about $43 million in Australia each year. And the main crops being affected are wheat, barley, oats, canola and lupins. Now, the nuisance factor of slugs and snails is well demonstrated in the panel on the right. This is an image of a header of a mower that's used to cut canola, and it's covered in thousands of greyfield slugs. And these need to be cleaned off and cause a real nuisance at the time of harvest. So what are the current options for control of pest molluscs? So one of the strategies is to reduce the number of molluscs that are retained between crops. Agricultural practices over the last decades have seen us move from tilling fields to no-till agriculture where the stubble is retained. Now this preserves soil structure and helps with water penetration. However, the remaining stubble provides a refuge for molluscs. They can either shelter under the stubble, or as seen on the right, they can climb up the stalks and rest there. So there's two ways to get rid of molluscs between crops, which is either to burn the stubble as shown here, or to knock them down during hot weather onto the hot ground, where they'll either die due to temperature stress or be crushed by a roller if you have one. In the garden setting, you're more likely to use a slug or snail bait, and these are known as chemical molluscicides. They're also used extensively in horticulture and agriculture. For example, in Australia each year, we apply about 3,500 tonnes of bait to canola crops, and this costs in excess of $12 million a year. The baits are essentially a pellet of bran with a dose of toxic chemicals. And the main chemicals are metaldehyde, methiocarb, and iron chelates. There are problems with these chemicals. They're toxic not only to the target species, the pest, but also to other animals. So methiocarb in particular is a problem with regard to off-target effects, as it can kill birds, mammals, and other non-target insects, which may be beneficial. This environmental risk of using methiocarb has resulted in it being withdrawn from use as a molluscicide in the European Union. Now, recent uses of methiocarb have also been challenged, principally because there have been issues with it getting into drinking water supplies. Other issues with baits is they often perform badly and have to be reapplied to the field due to degradation, or the pests not being actively feeding. The worldwide market for molluscicides is huge. Over $500 million worth of sales occur for chemical molluscicides each year, and the market is growing particularly in Asia. There are some non-chemical molluscicides which are categorized as biological molluscicides, although they may include living or non-living agents. So this market is worth about $50 million, and it's comprised of minerals, animal products. There's a nematode and for controlling slugs, and some bacteria for controlling muscle attachments. So this is the area that we wanted to look at to see if we could come up with new ideas in the biological molluscicides where we could find a way of controlling pest slugs and snails with limited off-target toxicity effects. This is a panel from a recent market analysis on molluscicides, and it points out some of the driving forces for market growth. There are the demand for high-value horticultural crops, which are particularly vulnerable to mollusk attack. There's the restrictions on using some of the toxic chemicals, and there's emerging markets in our part of the world and a desire to have some sort of eco-friendly green types of products that can be used. So we've got this growing awareness of looking at alternatives. I've decided to bring one of my acknowledgement slides up the front because it's really important in this story that it's a story of having partnerships. On the left, we've got Associate Professor Derek Russell, an entomologist and a very keen bird watcher. Now, he's responsible for getting me into the slug business in the first place. And I'd have to say it's been a huge pleasure to work with Derek over the years. All the research I'll present today is from projects that Derek and I carried out with the Grains Research and Development Corporation. I'd like to acknowledge the enthusiasm and support of Ken Young and Lee Nelson from GRDC. They've been on this journey with us. This is the front cover of a recent GRDC publication, which featured a photo of the protozoa that we work with. And I'll describe that later in my presentation. But let's start with the first idea that we worked with GRDC, and that was RNAi. So Derek and I worked on a project with GRDC on a proof of concept looking at whether it was feasible to use RNAi for slug control. The goal was to try to include RNA into a slug or snail bait, which was lethal for the target species, but was not lethal for non-target species. This is a simple scheme of how RNAi works. A double-stranded RNA can enter into a cell and the two strands separate. We get a single-stranded small interfering RNA. If the sequence of that RNA matches the messenger RNA produced in the nucleus, the two strands can hybridize. So normally this messenger RNA would get translated into protein to be used by the cell, but if it gets complexed with the small interfering RNA, it gets processed by a complex called the RISC process, or RNA-induced silencing complex. The complex produces enzymes to cleave the nucleic acids into small pieces, and it blocks gene expression by destroying that messenger RNA. If the gene was essential for the cell, then the gene silencing can damage the cell. Now, scientists studying nematodes discovered that they could design double-stranded RNAs against specific genes, and they then went on to develop a large body of work of using gene silencing to try to combat various pest insects. Here we see a scheme from a recent paper showing a number of different ways that double-stranded RNA could be delivered to control pest insects. The ones with the blue spots, such as here, are suitable for large-scale application to crops. Things like transgenic plants, baits, foliar applications, and seed dressings, and even recombinant viruses. Interestingly, it's only just last month that the first commercial RNAi product was approved. And these are transgenic plants called smart stacks, And they are corn which have got RNAi in them, which can control the corn rootworm. And there's a lot of other products in development, including some of the RNAi type sprays with this technology. So we wanted to look at whether we could deliver RNAi through baits or by sprays, so that we could get slugs or snails to ingest a dose of RNA that would either inhibit their feeding or inhibit their survival. When we started this project, we were really encouraged by a report that came out from Maddie Knight and the group, where they showed gene knockdown in this snail, which is Biopesticides glabrata, and it's the host of the worms that cause schistosomiasis. This paper was a breakthrough and heralded a new era where we might be able to use RNAi to combat the mollusk vectors of parasites. So we turned our attention to the grey-field slug, because this is one of the very big, one of the major damaging species. We needed an idea of which genes we could silence, and to address this, I need to do a little bit of slug anatomy here. So we've got the diagram here with the slug and its digestive tract. The major organ down at the base is called the digestive gland. And this is carries out the roles of a pancreas and a liver, and it's the site of major enzymatic digestion. There was a preliminary transcriptome of the digestive gland available, and we mined that data set for candidate genes for RNAi. The genes were guided by what was working in insects, and we started with conserved genes and went on to look at species-specific genes. Once we had a list of genes, we needed to put a lot of other systems in place in order to test those candidate genes. We developed systems to produce that double-stranded RNA. There's a gel of some purified double-stranded RNA here that was expressed by bacteria. We also used chemically synthesized RNA. We needed a plentiful supply of slugs, and consequently became quite skilled in breeding slugs. And that's because the animals that we wanted to use needed to be readily available year-round. We had a number of different formats that we could incorporate the double-stranded RNA in, including nanoparticles that could be incorporated in cereal baits or applied to leaf discs, as you can see down here. And we also needed to have techniques to track RNA in animals and measure its stability. The outcomes we were looking for were mortality or a reduction in grazing. Up here you can see a couple of our students, Constance and Craig. So once we had all the systems together, we started testing survival of individuals. We had some promising results using egg injection experiments. So this is just a laboratory scale technique. On the right, we can see a slug egg. It's got a jelly-like shell, an albumin, and there's the embryo, and they're transparent. So they're quite easy to inject double-stranded RNA into this albumin. And we did that with a number of targets. In the graph here, we can see the controls are in yellow, and two of the targets, one singly and one in combination. And we got lower survival of the embryos after hatching using some of these targets. But this is a laboratory technique. We wanted to apply it to ingestible baits. Disappointingly, this is the sort of data that was typical of the assays, where we measured mortality from feeding. Here we put double-stranded RNA on leaf discs and had an assay system like this. On the vertical axis is the survivors, the number of survivors out of groups of 20 animals, and the bars in green are our controls. And the bars in blue are various target formulations. And what we saw is all groups survived well. We were concerned that perhaps our RNA was degrading in the bioassay system. Perhaps it degraded on the leaves or in the baits, or was digested by the slugs before it reached the target organs. We did quite a lot of work on and had some nice experiments showing that the RNA was actually very stable in our assay systems, and it did reach the target organ. This shows a dissected slug. This is the skin, and here we have the crop. The mouth is up there, and the digestive tract runs down here and wraps around the digestive gland, which I've outlined in yellow to highlight it. So this is a slug that was fed fluorescently labelled RNA 24 hours before it was sacrificed. On the right-hand side, it's the same slug, but we've taken the photo under a wavelength that excites the fluorophore that we use to label the RNA. And you can see that the digestive gland is glowing red. And this demonstrates that the RNA got all the way into the digestive gland, reached the target organ, but we didn't see any gene silencing. So for gene silencing to occur, we need all the components of that risk complex to be present and operational. This has been a very quick summary of a lot of work and a lot of effort by a fairly large team. We were left with the question as to whether RNAi could actually work in slugs by ingestion at all, and whether it could spread systemically from the cells that first received it and affect whole groups of cells and organs. A recent review of progress in the area is summarised in this table. There's a list of half a dozen species of molluscs, and I've illustrated them down in these pictures here. And to start with, it really emphasises how molluscs are a very diverse group of animals, and there's lots of lines of evolution. So collectively, molluscs is probably simplifying the biology enormously. If we look at the methods that the researchers have used to get gene knockdown or gene silencing, we can see that they're laboratory methods, soaking of larvae or injection. So we did have some promising results using these types of techniques, but it's a long, long way from actually delivering a double-stranded RNA in a bait. So the current opinion is that some, but not all, molluscs have RNAi, and the system in molluscs differs substantially from the well-characterised systems in insects. So we talked to GRDC and decided to pivot to a new idea. So we wanted to look at a different biological alternative, and this stemmed from our early days of setting up a slug breeding colony. We had a lot of problems getting the eggs to hatch and to get the neonates to survive. Now, the colony was a really central resource, so we put a lot of effort into refining our methods and re-established the colonies for most of the RNA work using eggs that was screened and cleaned and ensured that they were healthy to establish new breeding lines. But I kept wondering whether there was some microorganism responsible for the deaths of those early eggs and neonates. Now, I'm a microbiologist, so I did what microbiologists do and have a really close look. There we go. So under the microscope, this is the sort of images I could see. On the top left up here is the body of a slug and all the moving objects of protozoa that are consuming the slug. We wanted to hunt down this organism and see if we could use it as a biocontrol agent. So we proposed to GRDC that we do a proof of concept project to identify the organism and test out their capability. So Derek and I did a large number of examinations of slugs from the wild, and we found a whole variety of different protozoa. We're particularly interested in some protozoa that we observed inside slug eggs. On the left, we can see a normal healthy slug egg, and in the middle and on the right, there are two eggs which have got these spots in it, and we could see those swimming around within the albumin. This right-hand egg actually has the embryo being consumed by those parasites. This one, it's still surviving. Now, we actually used some eggs like this to extract and purify the parasites. We identified it as being Tetrahymena rostrata. So this is a scanning electron micrograph of a cell. They're about 20 microns across and 50 microns long. They are a species that had not been previously reported in Australia. They're a single-cell eukaryotes that are covered in these rows of cilia, which allow them to swim through liquids. You can see that they, from this angle, is sort of the back of the cell. On the other side, around about here, there's an oral pore, and that has rows of cilia in it, which are arranged like an eyelash, and they beat backwards and forwards and sweep food into the mouth. This is what we thought was causing the slug deaths. So we purified it, and we needed to be able to test that. So we established some systems where we could grow up the organisms in scalable culture. So using a little bioreactor where we can control pH, temperature and aeration, we were able to use very cheap and simple media to get about 10 to the 6 cells per mil. This type of format can be scalable up to the hundreds of litres, potentially thousands. That's a whole area of development which we're now really looking at. So having grown organisms, and we started looking more at and understanding their life cycle. There are 3 different developmental forms. The ones, like I've already showed you here, these are called trophons. They're the feeding form. If the environmental conditions are unfavourable, such as through starvation or stress, they can convert to a cyst, which is shown in the middle. And when conditions improve, cells emerge from the cysts and have another feeding form called a theront, which are very fast and aggressive. And you can see them moving around a bit of slug tissue on the right there. So I'm going to use the cartoons shown below to illustrate trophons, cysts and theronts as we go along. Interestingly, theront really means wild beast. They're very hungry and aggressive. So we wanted to actually start with our trophons. We could grow them up in a flask or in a culture and generate large amounts. The life cycle essentially would go trophons through a few steps to form cysts, where we would have excystment theronts, which convert back into trophons if they get a food source. So one of the strategies that we were thinking about doing is wondering whether we could actually get the cells into this cyst form, which could be stable enough that a product would have enough shelf life that it could be manufactured, packaged and transported to growers for use in the field. But we started off looking at actually the virulence of the trophons. So my first question was, we could grow up the trophons easily, could we kill slugs? On the left is an assay where we exposed adult slugs to trophons and measured mortality over time. The green line is the survival of the unexposed slugs, and the red line is the progressive death of the group of slugs over time. And we were really encouraged by this experiment. We'd spent a long time not killing anything with RNAi, and then this was the first data that we had that we might actually be able to have a biological molluscicide. The slugs we used in this assay were reproductive age, and we actually looked at the impact of the next generation. If we collected eggs around about here before any mortality, We can see both the controls and the exposed ones laid plenty of eggs. Most of them hatched, but the survival of the neonates in the exposed group were dramatically different. And we thought, okay, perhaps the juveniles are particularly sensitive. If we compare the susceptibility of adult slugs, I've shown this graph before, and juvenile slugs, you get the same basic pattern, but the time scale is different. Juvenile slugs die a lot quicker. So this became our target life form of the slugs. If you could get T. rostrata into the soil where it could kill newly hatched and young slugs before they got up into the plant canopy, then we might actually have a control strategy. So most of the work going forward was done on juvenile slugs. We started developing techniques where we could manipulate the life cycle and get trophons to encysp, exist, and produce therons. So our next question was to compare the different developmental forms. And we set up some experiments where trophons were starved, then existed and released the therons, and we used those therons in bioassays. So again, the results shown here with the green line showing the survival of the unexposed slugs, and the blue line is the deaths of slugs exposed to trophons. And the main difference is if with the therons we had this big drop in survival in the first week, and thereafter the death rate was similar in both of the exposed groups. This has been fairly reproducible, provided that the cultures used to make the therons aren't too old. Now, trophons do kill slugs, they just take longer. And you need to remember that therons turn into trophons once they get a good feed. So this was encouraging, and we started on methods to efficiently produce and store cysts so that we could have a product that was primed and ready to release the infective therons. This graphic summarizes a huge number of experiments that Ruth Height has performed to optimize the growth conditions and the physical conditions to get good encystment. The beakers represent all the different growth conditions. Flasks. The beakers are the different sort of tests that she did to work out the right conditions to get successful encystment. Our criteria for success was that we wanted high density growth in the culture phase, and we wanted synchronized encystment, very high efficiency conversion of trophons to cysts, and a high level of survival of cysts that could release therons. The best processes could result in almost 100% encystment within 24 hours. And you can see some of the data in this graph where we see the increase in cysts over time. I'll comment more on the variables on encystment shortly, but I want to show you a few characteristics of the cysts. What we see when cysts form is they start getting a coating on the outside of the cell. This is a section and a transmission EM of a cyst. We start getting this wall building up. And this is through an extrusion of some mucus from these organelles called mucocysts. Eventually the cilia, and you can see some remnants of them there, they stop beating and the cyst becomes sticky and will attach to a surface. On the right, we've got some pictures of cysts that are spinning around in the capsule, and this one down on the bottom left is going to pop out of its cyst capsule. This is encystment in real time. On the left is a still image of a whole lot of cysts sort of attached to a surface. Some of the things that we found that encouraged efficient encystment was providing the right sort of solid supports. And all the brown material on the left are some particles that we add to encourage good aggregation of cysts. So we had success, yay. But there was a problem. Although we had a good method for efficiently making cysts that we could store at room temperature, we encountered significant problems with storage. On one hand, they were viable, but they spontaneously existed, or in other conditions, they lost viability. We tried a lot of variables and it was a big challenge. So how could we stabilize the cysts? So we're looking at getting cysts from this form into this form. A cyst that will spontaneously exist to a cyst with a long shelf life. The difference is being in a cyst with a thin wall through to ones like this that have got a thick wall. They're much more stable. These are again transmission EM images that Liliana prepared. We tried a number of different formulations and one of the surprising results was that encapsulation of the Tetrahymena in hydrogels prolonged their shelf life enormously. Samira Seracina did some of these assays where she encapsulated trophons in calcium alginate beads. As the calcium cross-links the alginate during gelation, the trophons were actually driven to the center of the bead. The surprising thing was that some of them formed cysts in the process. We could pre-make cysts by starvation and encapsulate them. And again, we were surprised that encapsulation stabilized them. So rather than either dying in a week, they could remain stable at room temperature for months. We're able to incorporate some compounds that attract slugs. These are some pictures of slugs having a munch on some gel beads that we made that have got a carboxymethyl cellulose center and an alginate shell. At this point, we had a number of processes for growing, encysting and storing cysts. The main variables for efficient encystment were the concentration of nutrients and cations, the pH, the encystment and storage temperature, and the time. We started to talk to our colleagues. Sorry, let me just go back. We not only had encapsulation as a way of stabilizing cysts, which I've already showed you, but certain buffers could stabilize them and dehydration under a certain chemical formula. So we had a whole lot of processes, and so at this point we started to talk to our colleagues at Research, Commercialization and Innovation about the commercial potential of the technology. And in particular, Lachlan Wilson and Greg Harper have become important members of a team as we progressed to protect the IP by lodging provisional patent and start conversations with industry to explore the possibility of taking the technology from the lab through to the market. I can talk more about that process with you, but at the moment I'll focus a bit more on the data and perhaps we can return to this in question time. So we've always got our eye here on the market. So this is an investment by GRDC, and we're always mindful that they have stakeholders and they want to see a return on investment. So how would you take these types of discoveries and take them to market? We're only in the discovery phase of an invention, and there's a long, long way before you might see this as a product on the shelves in Bunnings. In Australia, any product such as a molluscicide from Tetrahymena would fall under the authority of the AVPMA for registration. And here's a list of all the things that you'd want to have data on, or they would want to have data on before approving a biopesticide such as Tetrahymena. Now, some of these things we have quite a lot of data on already, and others will require new projects to satisfy the regulatory requirements. I want to just talk a little bit about a couple of these points: the site of infection, the mode of action, and the host range and specificity. This is some work by Anne Watt. We had observed during our challenge experiments that there was a behavioural change in slugs prior to them dying. They would start having difficulty moving and they stopped feeding. And Anne did a study looking carefully at all the signs of infection prior to death. She categorised slugs as healthy, ones that had mild impairment of their ability to extend the head and the eye tentacles. And this went from a mild to a quite disabled slug. And this one, death is imminent. And what we see is if it took 12 uninfected controls and observed them every day and scored them green as healthy, orange is whether they had these eye tentacle problems or dead, we see maintenance of good health in uninfected slugs, but each of the exposed slugs go through having an eye tentacle impairment phase followed by death. So Anne infected a whole lot of slugs and collected those early before they died. We're now showing these early signs of infection to see if she could determine where the parasites were. Now, this is one of her beautiful cross sections of the slug, and we'll need to address another slug biology 101 in this slide. This section of the slug, I've tried to show where it's cut through the slug here, so it's down the midline, and this is the orientation of the slug with the head to the left and the tail at the right. The main area of interest is up here, which is where the shell would be if this was a snail. So on a slug, there's a flap of tissue called the mantle. And underneath that mantle is a remnant of a shell. It's a sort of evolutionary remnant. It sits in a cavity called the shell cavity. And that protects the cardiopulmonary cavity. And inside there we have the kidney and the heart and the lung, and they're sort of all intertwined. The digestive gland is down this end, just for reference. And it's really large and it's all sort of intertwined with the crops here and digestive tract. So this is a healthy slug, and exposed slugs to Pterostrata theronts waited until they showed signs of infection, fixed, sectioned and stained them. And here's the sort of thing that she saw. So again, we're looking at this part of the slug and the section is through here. What we can see through this section is there's the mantle, there's the shell cavity, and here's the cardiopulmonary chamber. I have outlined for you all of the Pterostrata cells. The vast majority of the cells were in the kidney. A close-up of just this area is shown over here, and the cells here, these are all Pterostrata. You can see the little cilia on the outside if you could look closely, and they're interfering with this epithelium of the kidney, and they're causing damage to those cells. In severely infected animals, there's substantial damage to the renal tissue. And we think that the behaviour that we see is that the animals are having trouble with managing their water balance and they're dehydrating. And was also able to see some of the parasites in the heart and lung. And in severely affected animals, you could see fewer in the other tissues. Ultimately, though, an infection ends up in just liquidizing the whole animal. They just digest it. Interestingly, there was no evidence that the ciliates were just burrowing in from the outside, which raised the question of how they're getting into the kidney. So the potential routes of entry are shown in this section. And again, we're cutting down here through the slug. We've got the mantle at the top, the shell cavity. And in this slide, which is an uninfected animal, we've got the lung, the kidney, and the heart. Now, the design of a slug body is that there's a gap in the mantle called the pneumostome, and this is the route through which slugs breathe. And so we end up in this area with the lung. And close by, underneath a flap of the mantle, is the excretory pore, where the waste products go. And there's another point of entry, and there's a little hole just underneath the back dorsal flap of the mantle. And that's called the integumental pouch. And that goes into this shell cavity. Now, there aren't any other holes to get in to a slug unless the parasites bored through tissue. We've never seen any parasites in the shell cavity. And I think they probably enter either through the pneumostome or the excretory pore and then swim up into the ureters and get in through these loose connective tissue. On one side of some of this tissue is kidney cells and the other side is respiratory epithelium. So I think what is happening is that the ciliates are being attracted to nutrients that are available in the kidney. Most parasitic protozoa can't make purines. They have to scavenge them from the environment, and slug excretory fluids would be very rich in purines as a source of nitrogen for the ciliates. What about host specificity? The picture on the right is a collection of slugs that Michael Nash came in from the field with one day. And you can see there's a large variety of slugs that he got from this particular field. Now, will T. rostrata kill everything? No, it won't. We have tested the susceptibility of a number of slugs, including all the ones that are shown on this plastic lid. Some are susceptible and some are colonized but didn't die in the tests. So on the left, I've listed some of the ones that are susceptible, and the black-keeled ones, I said, were colonized. So out of this tray, the ones that are susceptible, well, the ones that may not get killed are indicated with these spots. So you could actually have a pretty good impact on the slug population, although variable impacts on different species. We've tested garden snails that don't seem to die, although some get colonized. We've tested some sort of worms that are resistant, and we've got tests ongoing on a number of the pest snails. We're going to have still a lot of hurdles in scaling up and formulating materials so that we can actually do real field trials, but so far the results are very promising. I've listed here the contributors to the project over the years, including people who have been in the RNAi phase, people who've done field collections for us, a number of students, and of course our colleagues in RIC. And I also wanted to acknowledge Simon and Andrew, who are in the translating research at Melbourne unit. They are they're part of a terrific team that can give people a lot of help in thinking about their research as being a product, and how people can think about prospective customers and examining the pathway from the lab bench into the field. Now I've got one. Come on. Here's a bit of a mugshot of some of our team. Up here is Derek, who is happily retired. Down there's Michael, who introduced me to slugs. Kerry Dunce and Chris Lum and Yuhong Liu and Constance and Craig were all involved in the RNAi phase of the work. Denya Heap and Anne Watt and Samira Sirasina have been involved in the protozoa work. And I want to make special mention of Ruth, who is a major driver of the laboratory work and experimental designs. And Mike Dale-Smith, friend and colleague, who's joined me on a number of field trips collecting slugs, which is always fun. Here are the guys from TRAM, And many thanks to Lachlan, who's guided us through all of the IP and commercialisation steps along this journey. I thank you for your attention today.
Helen, thank you. Thank you very much for that talk. That was fascinating. I've got a lot of questions, but before we launch into questions, can I just ask everybody that would like to ask a question, if you could put it into the Q&A tab and not the chat tab, please. Put it in the Q&A, and you might get to ask the question live, or I can ask it for you if we can't get through to you or you don't have a microphone. So yeah, put the questions in the Q&A, please. Helen, maybe I could just start while people are doing that. And, you know, the protozoan that you're using there seems to be a bit like Alexander Fleming and a sort of serendipitous finding that you suddenly realized your slugs were dead and you came up with this protozoan that was killing them. And you've followed that, and it's been very successful. But, you know, is there any value in looking for other protozoa, actually getting out there in the field and seeing what other protozoa are about? Because there might be some that are perhaps even more virulent to slugs.
Yeah, it's a very good point, and we have a number of those protozoa in cryo storage, and we've got other isolates of this species, and it's really a workforce issue. So while we were developing ways to handle this one, it was just not feasible to do large numbers in parallel. But we have all our systems so refined now that we have started looking at other isolates, because we can put them in a slug assay and we can knock down slugs in a few days in a bioassay. So we've got a much higher throughput system for testing different potential parasites, and it's something we're quite excited to do more of.
Okay, thanks, Helen. If I look at the Q&As, the first couple of questions that I see are asking about the specificity of T. rostrata. How specific is it to slugs or other mollusk species? And I think you addressed that, and these questions were sort of asked, I think, perhaps before you got to that bit of your talk. And then there's a question, have they been tested against any native mollusks? And I think you addressed that, because the lid of you showed us covered in slugs. Those, I think, were all from the field, weren't they?
Well, they're all invasive species. The native mollusks is a relevant question, because you don't want to knock off the beautiful Otway red snails or something like that. We've done a study and prepared a report on the distribution of the native animals and where people are cropping, and there is essentially no substantial overlap. So like what we're putting snail baits on the fields at the moment, they're in agricultural settings, and the opportunity for getting into native animals is not as high in those large usage areas.
Okay.
I think slugs are going to be more sensitive than snails in the long run.
Okay, thank you. Marshall Lightowl, Marshall, do you have your microphone set up? Would you like to ask your question?
Hi, Helen. Thanks very much.
Marshall.
Terrific talk. My question is, what minimum percentage of killing and rate of killing do you think would make the product commercially viable?
50% kill or 50% reduction in grazing are the targets that we've worked on. And they're the targets that we've talked about with GRDC. So if you could knock down the damage by half, then you're on a good thing.
And if it takes 8 days to do that, is that still okay?
What happens once you get one slug infected, then the organism amplifies up and infects the next slug. This isn't going to be a quick knockdown effect like you throw out your baits on a rainy night. it's more managing population in the soil. You want to knock down the soil load of the eggs and the young ones. So it's going to be required forward planning rather than be reactive on a rainy night.
Okay, thank you.
Thank you, Marshall. Andrew Fisher, would you like to ask your question? I like this. It sort of follows on from some of the other things. Andrew.
Yes, thank you, Helen. Fascinating. I never probably found slugs so fascinating since I was probably about 2 and ate them in the dirt. But my question is relating to the biological control strategy you're using. I guess I was wondering, do slugs do any good things? And is there any risk to that from unleashing the sort of biological control that you're developing?
Slugs do good things. Their part in the environment is to recycle plant matter. So they break it down, put it back into the soil. So they do have a good role. The problem is when you're trying to grow a crop and you've got those thousands and thousands and thousands of an invasive species destroying that. And so trying to get a balance is the thing. And I really think the farms of the future are looking at these types of things as part of an integrated pest management strategy. There's not one solution. Choosing crops, matching season, using an armory of tools is really the solution to balance. And that's where we see this as being one part of a more complex control option.
Thank you.
Thanks, Andrew. Vern Bowles. Vern's got a question for you, Helen.
Hi, Vern.
Hey, Helen, how are you?
Good.
Good. Yeah, just I'm interested in the your perceived MOA with this. You've got some slug species that you think are refractory potentially to the treatment. I want to know, based on your MOA, can you explain why that may be the case? Because I'm assuming slug physiology is relatively homogeneous.
Well, actually, it's not.
Good.
The black keel slug, which we so far haven't been able to kill, it may simply be it needs a bigger dose. They're a burrowing slug, and I think that some of the points of entry are a bit smaller.
Mm-hmm.
And I think we've got to do some more work in that area, and it's a bit like John says, that if we've got a panel of different protozoa that we can, and we've got a number that can go through encystment, and we can put the same things in. Perhaps matching the target species to the parasite might be an approach we could take.
Okay, and just on the physiology, you said that actually they're not that similar. Could you comment more on that?
The integumental pouch in the black keeled slug is either not there or it's very small. So if that was the primary point of entry in the grey field slug, it just may be that they can get into that more susceptible species easier.
Okay, thanks.
So fine details of entry.
No worries, Tar.
Thanks, Vern. The next one says anonymous attendee, so I'll ask this one, Helen. So the question was, the market in Australia might not be big enough to cover the cost of regulations of actually launching this as a product. Are there global markets, and where might these be most suitable? And I would probably add to that, will there be a resistance to taking a protozoan that is not from, let's say, Africa, if people tried to use it in Africa or people tried to use it in Europe? Or is this protozoan found globally?
Sure.
So what is the global potential for this, Helen?
Okay, I regret now deleting a certain slide. The distribution of the slug species coincides with the natural distribution of T. rostrata. It was first described in 1926 in Germany. And so where it occurs and where the pests occur are covered the North American and European market, Australia and New Zealand. South America has the pest slugs, but we don't know whether the ciliate is there. So there is the potential to isolate relevant strains in the jurisdictions if that's a barrier.
Okay, all right, thank you. Robert Day is asking a question, but I think we've addressed that one, Robert. It's about how the resistant species are resistant, which I think is what perhaps Vern was asking. I might have misunderstood that, but I'll move to the next one. Sarah Franklin asks, for the animals that were colonized but not killed, did they show any reduction in grazing? Do they eat less? You know, is that effective? Do they have to die?
No, well, so it's a good question, Sarah, because those black-keeled slugs, the 2 assays we've done, we just looked at grazing and death. There was no impact. But I'd like to actually take those through to see what the next generation effects are. Does it actually reduce their fecundity? That's a longer term experiment because they grow a bit slowly, but it's something that we can certainly do. We actually have a black-keeled slug colony upstairs at work, not here.
And then I have one from an anonymous attendee, and there might be intellectual property associated here that you don't want to talk about, but do you have companies interested in taking this forward?
Yes.
Okay. All right. Well, that's good. So that's nice to know.
And look, really, this is where Lachlan Wilson and Greg Harper and Robert Mann have been enormously helpful to us. And we've spent a lot of time preparing and presenting and discussing potential with industry, and we've got some strong interest.
Helen, if I might just come to a last question, because we haven't really had any questions about this part of your talk, and that's on the RNA work that you were doing. I mean, in recent, well, actually only in the last 12 months, Pfizer and BioNTech and Moderna have brought to everybody's attention that you can encapsulate working RNA molecules, let me put it that way, and deliver them successfully, in their case, vaccines for COVID-19. But do you see a future for the sort of RNA technology in control of slugs? I mean, it's being used quite a bit in insects.
It has been, yeah. and there's a whole suite of experiments I would like to do. Yeah. We just need to get some funding to do those. So I think the question is still open. And yeah. Since we did our first gene target analysis, there's been improvements in the understanding of the genome of the target species, and we've got much better tools, and there's definitely potential there that should be exploited.
I think also there are alternatives that you might want to consider, and I'd love to have a chat to you about this at some point. But you know, you could imagine putting in bits of RNA that would only be would have a promoter that was a slug-based RNA promoter and would produce a toxin, or self-replicating bits of RNA that would amplify themselves. Or you know, you can do that kind of technology now. We're using that approach a little bit in my lab in regard to insects, and but yeah. I think there's a lot of possibilities there.
When I first started that work, I'd really thought, I was surprised how robust the double-stranded RNA was. We could get it, we could put it in digestive juices and of slugs. We could put it in soil and bean leaf assays and things, and it was really very stable. There's some basic biology about RNAi in slugs. And because that part was sort of basic knowledge building, is why we went on to this other approach, because the GRDC were keen to look at sort of getting something in field.
Yeah.
But yeah, I think RNAi still got some legs to it.
I suspect it does. Yeah. Helen, I think we've reached the end of our time, and there may be other questions, but I'm sure people can email you with them. And I'm sure a lot of people will watch this later online. All of our Dean's research seminars are available online through the faculty's website. So Helen, thank you very much for a fascinating talk. I really enjoyed it.
Thank you very much. And thanks everybody for attending.
Thanks. Bye-bye.